In 1953, science was revolutionized with the critical work of a physicist, a biologist, and a chemist. But to understand the significance of their work, we first have to take a page out of the nearest biology coursebook you can find and turn to the topic DNA.
What? Oh, Mr. DNA, where did you come from? From your blood. Just one drop of your blood contains billions of strands of DNA, the building blocks of life. A DNA strand, like me, is a blueprint for building a living thing.
So let's start off with first getting down the definition for DNA as Wikipedia classifies it. Deoxyribonucleic acid is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth and reproduction of all known organisms and many viruses. A bit much, so let's define it in layman's terms by stripping some information off. DNA just stands for deoxyribonucleic acid, so let's just substitute that in. So that'll make it DNA is a molecule composed of two polynucleotide chains that coil around each other to form a double helix. That's still a bit of a mouthful really, so let's change that to DNA is a molecule which consists of a double helix with nucleotide bases. Now let's take a look at the rest of the sentence. Perhaps we can change carrying genetic instructions and so on to with sections called genes that contain the coded information for making proteins. Proteins, as you remember, are sometimes enzymes which control cells' chemical reactions, and just add a couple of words here and there, and there it is, our definition for DNA. DNA is a molecule found mainly in the nuclei of cells consisting of a double helix with nucleotide bases. A short section of DNA is called a gene, which carries the coded information for making proteins. So now that we've learned a bit about DNA, let's take a stroll down memory lane to learn more about the structure, which was the most unprecedented discovery perhaps of the mid-20th century. The tale goes like this. Two scientists combined together the findings from a vast range of other geniuses like themselves to come to a single model of DNA. These two revolutionary minds belong to James Watson and Francis Crick, winners of the Nobel Prize in 1962 for their achievement. The two scientists worked together at Cambridge University and were the first to cross the finish line in this scientific race, with others such as Linus Pauling, who discovered protein secondary structure, also trying to find the correct model. Rather than carrying out new experiments in the lab, Watson and Crick mostly collected and analyzed existing pieces of data, putting them together in new and insightful ways. Some of the more crucial clues to the DNA structure came from Rosalind Franklin, a chemist working in the lab of physicist Maurice Wilkins. Rosalind Franklin had been researching the structure of a number of substances using a technique called X-ray diffraction. When once the crystallized form of a molecule such as DNA was exposed to X-rays, some of the rays were deflected by the atoms in the crystal, forming a diffraction pattern that gives subtle hints about the molecule structure. Franklin's crystallography gave Watson and Crick important clues to the structure of the DNA. Some of these clues came from the famous image 51, a remarkably clear and striking X-ray diffraction image of DNA produced by Franklin and her graduate student. A modern example of the diffraction pattern produced by DNA is shown on screen now. To Watson, the X-shaped diffraction pattern of Franklin's image immediately suggested of a spiral double helix structure for DNA. Thus, with Franklin's results along with other materials, Watson and Crick proposed the now famous double helix structure for DNA. In 1962 thus, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Medicine. Unfortunately, by then Franklin had died of cancer, and Nobel Prizes are sadly not awarded after death. Hence today everyone knows the renowned picture of a strand of DNA. But what does it actually compose of? A molecule of DNA is made from two strands of molecules called nucleotides. Each nucleotide contains a sugar molecule, a phosphate group, and a nitrogen base. The four types of nitrogen bases are adenine, thymine, guanine, and cytosine, and each two nitrogen bases combined together are called complementary bases. Now, complementary bases always link and bind together with each other with hydrogen bonds and never with any other base. This phenomenon is called the base pairing rule, discovered by again Watson and Crick, and this explains Chargaff's rules. However, one false conclusion amassed by Watson and Crick in their original model was that they inaccurately proposed that there were two hydrogen bonds between the bases of each pair. So what we know today is that guanine and cytosine form 3 hydrogen bonds compared to adenine and thymine forming 2 hydrogen bonds. Yet still, it perhaps goes as an understatement to say that the structure of DNA unlocked the door to understanding many aspects of DNA's functions, such as how it was copied and how the information carried was used by the cells to make proteins. Watson and Crick's models ushered a new era of discovery in molecular biology, and the model and discoveries that it enabled now form the foundations for much of today's cutting-edge research in biology and biomedicine.